Longevity. It's a word that's sometimes used as a synonym for life expectancy. But today we're going to take a little bigger view of that. When we look at life expectancy, we tend to look at that time between birth and death. But we want to break this up a little bit for today's presentation. Because in life, we spend about the first third of our life learning. This is when we're in school preparing for work and moving on with our life. We spend that second third of our life serving others, whether it's actually working that job that we're trained for, in a relationship, raising children. But today we're going to take a look at the third, that part of the life where we get to take a trip of a lifetime, where we get to go to the weddings of our children and our grandchildren and be part of those celebrations and to be active, and to see the birth of our grandchildren, our great-grandchildren, and to be part of the family that raises them. Today, when we talk about longevity, this is what we're talking about. We're not just talking about longevity in the fact of getting more days in our life, but we want to take a look at how do we get more life in our days. And this is what we want to begin to explore. When we look at the literature, what we see is longevity is something that's been studied for a long time. In this article, we see a reference to a book written in 1550, The Art of Living Long by Luigi Canero. And he talks about individuals who are not destined to die at 60 or 70, but with care and good constitution, we could live extremely long lives. Now, he attributed this to a principle of moderation in all things. But what we'd like to do now is take a look at what the science says. When we take a look at the science, the manipulation of caloric restriction is an experimental manipulation that's shown to extend lifespan in a number of organisms: yeast, worms, flies, rodents, perhaps even non-human primates. When we look at caloric restriction, we have to start to ask the question, can we starve ourselves to immortality? When we start to take a look at the science, is what we see is that we can promote longevity through diet. And we look at this in multiple organisms all the way up to humans. What we see is that in humans, severe food restriction without malnutrition results in many of the same physiologic, metabolic, and molecular changes associated with diet restriction in animals. And when we continue to look at this, we can see that there are mechanisms intracellularly that contribute to the longevity. The diet restriction, whether it be done in limiting what we eat, when we eat, or how much we eat, will result in a different cellular effect. Today, we want to spend time looking at the cellular process of what we call autophagy. When we look at autophagy and longevity, what we see is that longevity-promoting regimens, including calorie restriction, the inhibition of TOR with rapamycin, supplementing with resveratrol or the natural polyamine spermidine, are associated with the increase of autophagy. And what we see is clearing the cellular damage by autophagy is a common denominator in many of the lifespan extending manipulations. There's lots of mechanisms for how autophagy results in the increased longevity, and we're going to continue to explore those in the presentation. When we take a look at the benefits of calorie restriction, we can also take a look at what's called intermeal or intermittent fasting and see the effect there on autophagy. Calorie restriction tends to increase health span, but its adherence in humans is poor. So what we found is that the study suggests that consuming 2 meals a day without calorie restriction could prevent the metabolic syndrome associated with overfeeding. We also see in the literature that autophagy mediates pharmacological lifespan extension with supplementation of molecules like spermidine and resveratrol. Deficient autophagy is directly involved in a number of pathologies, including neurodegenerative diseases, heart failure, myopathies, and many other chronic inflammatory states. Autophagy occurs at low baseline levels in all cells to ensure the homeostatic turnover of some of the long-lived proteins and used organelles within the cell. But we see that autophagy is upregulated well beyond basal levels when cells need to metabolize intracellular nutrients, as occurring during glucose or amino acid deprivation, calorie restriction, hypoxia that might be associated with movement or exercise, or other growth factor withdrawals from the cell, again related to caloric restriction and or exercise. When we take a look at anti-aging effects of supplementing some of these molecules, spermidine comes to mind. Spermidine is a polyamine, and polyamines regulate cellular growth and death. The decrease in polyamine levels with age could play a part in cellular aging. Polyamine supplementation may help lessen the effects of cellular aging. We also see that spermidine is a naturally occurring polyamine and has recently emerged as exhibiting anti-aging properties. Its supplementation increases lifespan and resistance to stress and decreases the occurrence of age-related pathologies and loss of motor ability. Spermidine is a simple little 9-carbon molecule with 3 nitrogens that is synthesized originally from an amino acid. When we look at polyamines in foods, what we see is that polyamines are found in all living cells, including microorganisms, plants, and animals. Polyamines can also be found in the food of both animal and plant origin. And an important source of polyamines in humans is breast milk and infant formula. In foods, spermidine and spermine are primarily naturally present, coming from raw plant materials and some animal tissues. Polyamines are ubiquitous in foods of plant origin, although their content and distribution vary depending on the type of food. Spermidine, present in all plant-derived food, is generally the predominant polyamine. The food categories with the highest contents of spermidine and spermine are cereal, grains, legumes, and soy derivatives. Wheat germ and soybeans stand out in particular in their contents. The variability of polyamine contents in plant-derived products can be due to different factors, including their origin, growing conditions, harvesting, or storage. In this sense, different stress situations of plant can affect the polyamine content. So that we see that the induction of autophagy by spermine can promote longevity. And in this article, we see that the administration of spermine, a natural polyamine whose intracellular concentration declines with human aging, markedly extended the lifespan of yeast, flies, worms, and human immune cells. In this article, they found that the enhanced autophagy was crucial for polyamine-induced suppression of necrosis and enhanced longevity. The data indicate that exogenous supplementation of spermidine can retard cellular aging in several species. Spermidine retards chronic aging and also rejuvenates old cells. So today we've wanted to talk about longevity, its relationship to autophagy, and how spermidine is a key element in this process. In later videos, we're going to explore the role of spermidine supplementation with improved immunity, enhanced cardiovascular health, decreased neurodegeneration, improvement in bone and cartilage health, improvement in brain and memory performance, diabetes and obesity, and improved muscle function.